Hello. Today we will talk about the basis of molecular biology. So we must talk about begin with about nucleic acid, the structure of nucleic acid and organization. So the overview we must talk about the central dogma of molecular biology. You can see that in this first reaction, the DNA make a new DNA. It is called replication. DNA make a new DNA. Why the DNA need make other DNA? To pass the information to the new generation. Okay? This second reaction is the DNA making RNA. This reaction is called transcription. The transcription is used to express some gene. When you talk about uh, messenger DNA, RNA, okay? And translation. In the translation, the RNA will be the base to make a protein, which happens in a ribosome outside the nucleus of this cell. Okay? Sorry. So you can see here uh, an organism must be able to store and preserve its genetic information, pass that information along the future generation. To pass this information, we will make a replication and express that information as it carries out all the process of life. Okay? So, the genetic information is stored in the base sequence of the DNA molecule. This information is used to synthesize all the proteins made by an organism, and not so proteins, but RNAs, uh, not so messenger RNA, but in the ribosomic RNA, and it's and it transport RNA and microRNA. Okay? Basically, a gene is a unit of DNA that encodes a particular protein or RNA molecule. This definition is complicated by our increased appreciation of the ways in which genes may be expressed. We will talk before about the recombination of the DNA, of the RNA. Okay? when DNA, when a gene will be expressed, the first stage in gene expression is called transcription. It involves a transfer of information found in a double-stranded DNA molecule to a base sequence of a single-stranded RNA molecule. If the RNA molecule is a messenger RNA, then the process known as translation. It converts the information in RNA base sequence to the amino acid sequence of a protein. When a cell divided, each daughter cell must receive an accurate copy of the genetic information. DNA replication is a process in which each chromosome is duplicated before cell division. Okay? Reverse transcription, we didn't talk about in the first, but it is reverse transcription which produces DNA copies of an RNA is more commonly associated with life cycles of retroviruses or RNA making DNA. Okay? Nucleotide structure and nomenclature. So, nucleic acid is assembled from nucleotides, which consist of three components: a nitrogen base, a 5-carbon sugar, its pentose, and phosphate. So we can see a 5-base carbon sugar. It's a pentose. When you have a hydrogen in carbon 2, you have the oxyribose. If you have a hydrocarbonate in the chromosome 2, you have a ribose. Okay? If the pentose is a ribose, the nucleic acid is RNA, ribonucleic acid. If the pentose is a deoxyribose, the nucleic acid is a DNA, the oxyribonucleic acid. So, we can have a purine which contains 2 rings in the structure. You show it before, you show it after. 2 purines commonly found in nucleic acid are adenine and guanine. Other purine metabolites not usually found in nucleic acids include xanthine, hypoxanthine, and uric acid. Pyrinogens have only 1 ring. Cytosine is present in both DNA and RNA. Thymine is usually found only in the DNA, and uracil is found only in RNA. Okay? You can see the purine. So you have 2 rings, okay? The most common found purines is adenine and guanine in the DNA and RNA. Okay? Nucleosides. Nucleosides are formed by covalent link and a base to a number 1 carbon of a sugar, and the number identifying the carbon of sugar are labeled with primes in nucleosides and the nucleotide to distinguish them from the carbon of the purine or pyrimidine base. Nucleotides are formed when one or more phosphate groups are attached to the 5 carbon of a nucleoside. Okay? So, nucleotide is a nucleoside plus a phosphate group. Okay? So you have in the first an adenine attached to a pentose. Okay? Okay. Ribonucleic acid. So you make adenosine. This pyrimidine cytosine, one ring, you have attached to a pentose. You have cycline, and this other ring attached to a guanosine, attached to a pentose, you make a guanosine. And the last one, you have this pyrimidine. This is an uracil. The uracil attached to a pentose. Always uracil will have a ribose. Okay? So you have 4 ribonucleic acids. Okay? Okay? The cytosine, the pyrimidine, thymine, and uracil. Okay? Nucleotides are formed when one or more phosphate group is attached to a 5 carbon of a nucleoside. Okay? You have this purine because you have 2 rings. Okay? It's an adenosine. You have a pentose carbon sugar attached to a phosphate group. So you have a nucleotide. Okay? So these phosphate bonds are high energy bonds, or nucleoside D and triphosphate are high energy components because of the hydrolytic energy associated with the acid anhydride bonds. Okay? If you broke these bonds, this phosphate, you release an energy. Okay? So the nucleic acids here is nucleotides attached one another to another. In this picture you can see a DNA because you have one phosphate bonds, you have a double layer. Nucleic acids are polymers of nucleotides joined by 3 to 5 phosphodiester bonds. A phosphate group links the 3 carbon of a sugar to the 5 carbon of the next sugar in the chain. Okay? In one phosphate, in a 5 carbon and a 3 carbon sugar. Okay? And here the same. Each strand has distinct 5 ends and 3' ends, and thus has a polarity. A phosphate group is often found in the 5' end, and a hydroxyl group is found at the 3' end. Okay? You finish the sequence with the 3' group in the hydroxyl group. So in this picture you can see the hydrogen bonds between the double layer and the DNA sequence. And by convention, the base sequence of a nucleic acid strand is written by 5 to 3' end direction, the left to right. Okay? In eukaryotes, DNA is generally double-stranded, and RNA is generally single-stranded RNA. Okay? So you can see again a more complex sequence. The two strands are unparalleled or in opposite direction. The two strands are complementary. Always with T, 2 hydrogen bonds, and G always with C, because we have 3 hydrogen bonds. Okay? Okay. Cytosine and guanine, thymine and adenine. Okay, so you can see adenine is a purine and the thymine is a pyrimidine, and cytosine is a pyrimidine and the guanine is a purine. So you have always the same number of purine and pyrimidine. The amount of A equals the amount of T, and the amount of G is equal to the amount of C. Total purines equal total pyrimidine. It's called Chargaff's rules. Okay? One example of use of this DNA sequence is anti-tumor drugs like daunorubicin and doxorubicin are anti-tumor drugs that's used in the treatment of leukemias, and they exert the effect by intercalating between the bases of DNA, thereby interfering with the activity of topoisomerase II and prevents proper replication of the DNA. Cisplatin, which is used in the treatment of bladder and lung tumors, binds tightly to the DNA, causing structural distortion and malfunction. So, come back to the Chargaff rules. The DNA is a double-stranded, and the number of purine and pyrimidines is equal, okay? And adenine equal is thymine. They have a bond, 2 bonds, and with guanine and cytosine, there is the same number is equal. Okay? So, if you disrupt the hydrogen bond between the base sequence, you can have the denaturation of the DNA. It results in the melting of the double helix into 2 single strands that separates from each other. No covalent bonds are broken in this process. Heat, alkaline pH, and chemicals like formaldehyde and urea, are commonly used to denaturate. Renaturation occurs after the denaturation, and if the agent uh, is left from the reaction, and the DNA can renaturation or reannealing. Okay, such renaturation or annealing of complementary DNA strand is an important step in proving a southern blot and performing the polymerase chain reaction. So you can see an example of denaturation because the heat, more heat, denaturation, and if you cool this DNA, you can reform the, the base or renaturation. Okay, the DNA will be reformed. Other structures like mitochondrial DNA and the DNA for most prokaryotes are closely circular structures. These molecules may exist as relaxed circles or as supercoiled structure in which the helix is twisted and around itself in three-dimensional space. Negatively supercoiled DNA is formed if the DNA is wound more loosely than in Watson-Crick DNA. This form is required for most biological reactions. Positively supercoiled DNA is formed if the DNA is wound more tightly than in Watson-Crick DNA. Topoisomerase are enzymes that can change the amount of supercoiling in DNA molecules. They make transient breaks in DNA strands by alternate breaking and resealing the sugar phosphate backbone. So you can see the examples of the division from the DNA, and you can see the histones, the DNA around the histones to supercoiled the DNA structure. Nuclear DNA in prokaryotes is found in chromatin associated with histones and non-histone proteins. So you can see the H2A, H2B, H3, H4, okay? Histones are rich in lysine and arginine, which confers a positive charge on proteins. Two copies of each histone, H2A, H2B, H3, and H4, aggregates to form the histone octamer. Okay? And link the DNA. The DNA is wound around outside of this octamer to form a nucleosome or a series of nucleosomes, is sometimes called beads on the striking, but it's more properly referred to as a 30 nanometer chromatin fiber, okay, or a thick chromatin. Okay, if you have a nucleosome from chromatin 2. Histone AG1 is associated with the linker DNA found between nucleosomes to help it package them into a solenoid-like structure, which is a thick 30 nanometer fiber. Okay? Further consideration occurs eventually form the chromosome. Each eukaryotic chromosome in G0 or G1 contains one linear molecule of double-stranded DNA. So the nucleosome contains two types of chromatin. Chromatin, more open and available for genetic expression. You can see here chromatin. And if you have heterochromatin like here, this is more dense, more condensed, and is associated with areas of the chromosome that are not expressed. You can see chromatin corresponds to nucleosomes 10 nanometer fibers is in white, and heterochromatin in black, more condensed DNA, mas more highly condensed. Chromatin is with characteristic of mitotic nucleus. Nucleus here is a nuclear region specialized for ribosome assembly, or RNA ribosome. Okay, thank you very much. See you soon.